In this episode, we're going to take a brief break from the controversies over whale regulations and wind farms, for the most part, to learn about some potentially world-changing discoveries regarding amazing properties of the lobster that very few people are aware of. We'll hear from one of Maine's leading scientific experts on all things lobster. Most people know him simply as Dr. Bob. And he, along with one of his devoted lab assistants, specifically a yellow lab assistant, set aside some of their precious vacation time on Frenchboro last summer to have a chat with me about the untapped potential of the lobster.
I'm Dr. Bob Bayer. I'm Professor Emeritus of Animal Veterinary Sciences at the University of Maine and Emeritus Director of the Lobster Institute at the University of Maine. My reason for getting into lobster science really was to preserve a way of life and also to try to enhance the income potential for the lobster fishery in whatever way possible by the research that I got involved with trying to solve problems. I worked on things like alternative lobster baits. Not there yet, but I'm still actually working with this in my spare time, and I've got a couple concepts that still aren't done yet. One of the first areas I got into was the actual physical health of lobsters. There was a disease in the lobster population that was referred to as red tail, bacterial disease, and it was endemic in the lobster population. We could take blood samples of lobsters, and what we would see time after time is 5 to 7% of those lobsters had this red tail bacteria. And those that had it would die. There's no question. There was a large mortality taking place in the wild lobster population, but when these lobsters got placed into tidal pounds or storage tanks for storage, it really took off in terms of the mortalities. And sometimes a lobster pound operator would lose half their lobsters. And it would happen quickly, and the lobsters would die. The time to death was shorter when the water was warmer. And the way this would spread was lobster that was infected would get broken open by other lobsters that are nearby, because lobsters are aggressive and can be cannibals. And that released millions and millions of these bacterial cells into the water to infect other lobsters. Well, I was able to develop a medicated feed that was approved by U.S. Food and Drug Administration and also Health Canada. And that was quite a process dealing with the FDA. They are very thorough. We got good attention, let's put it that way, because the people involved in the lobster fishery advocated. We had a congressional letter that went to FDA, and that got their attention. And this was adapted very, very quickly. Virtually all the tidal pounds were using it, and in a short period of time, this disease was gone from the tidal pounds, and it was also apparently no longer seen in the environment. And we still don't see red tail disease in lobsters today. So this may be one of the reasons why we have these bountiful catches. Probably somewhere 20, 30, maybe even 40% of the increase in harvest starting in the 1990s, going from 20 million pounds to well over 100 million pounds on a regular basis, may be due to the fact that we don't have this disease anymore. It is gone.
Eradicating an endemic disease is no small accomplishment. Beyond Dr. Bob's scientific prowess, the main reason that he quickly earned the respect of fishing communities was that it was clear from the start that he prioritized the well-being of the people who his work would affect.
It's all about people, really. The lobster is a means to an end. It's about impacting the lives of people involved in the lobster fishery, not just in Maine, but I work a lot with the Canadian lobster fishery as well. And one of the ways that I saw that we could enhance the income potential from our lobster is to try to get more from the lobster with value-added products. And the ones that my company, Lobster Unlimited, is working on have huge potential. We're taking throwaway materials from two sources. One is the blood from the lobster, and the other is the shell from the lobster that we are processing and deriving a number of different products. One is a patent for treating mammalian skin lesions, and we can effectively do this with a lobster blood extract using the compound called hemocyanin. The other covers the areas of antiviral and anti-cancer properties of this same material. We are throwing away 2 to 3 million pounds of this raw material every year. And if we could get it all, I mean, it does have huge medical potential against a number of cancers, and I see the potential of a COVID-19 treatment, not a vaccine, but a treatment, because this material has a number of interesting properties. One is it's antiviral, and we're dealing with a virus. But the other is it stimulates the production of what we call natural killer cells, which has impact on cancer and a variety of other disorders, but it also is a viral scavenger. And the data that I've read says that if you have COVID-19, you have a deficiency of natural killer cells. So this might be a way of bucking up your own production of natural killer cells. And I would do this as some sort of inhaler to get it directly to the lungs. So if we could get somebody interested in working on this, I think it'd be absolutely terrific. And the cancer-related stuff, there's a lot in the literature that says that hemocyanin has efficacy in either inhibiting or killing a variety of different cancers. A lot of this is in cell culture. We culture cancer cells, we expose them to hemocyanin, and we see that we can inhibit or kill them. So I think this is another direction. Another product that we expect to release very soon is the same compound, hemocyanin, as a vaccine carrier. Most of the vaccines, with the exception of the new RNA vaccines, are based on a carrier and an antigen. The antigen causes the immune response, and the current material that's used as a carrier it's the same material but comes from a different animal. It's a keyhole limpet, which there's not a lot of it. The total production of keyhole limpet hemocyanin that supplies the vaccine market today it might be 5 or 6 pounds a year. We could do that in a day. Now, we now have a partner with Lobster Unlimited. It's a company we've worked with for several years, and they deal in blood products from a variety of different animals, about 20 different animals. The name of the company is Lampire. They've been around for 40 years. They have biosecure farms in Pennsylvania. They've got laboratory facilities. They're good at interacting and interfacing with big pharma, which we certainly need. We're also looking at lobster shells, and we're taking lobster shell from processing plants. And by the way, about 60 to 70% of the Maine lobster harvest is processed. And when we talk about processing, we're talking about picked meat and frozen tails primarily. That's a lot. And we dry and grind this shell. Right now, you can purchase it in most states at Whole Foods as a plant food, but it has other potentials as well. We're looking at making medicinals based on a derivative of lobster shell called chitosan. It's not an easy process, but we're doing this with a partner, Coastal Chitin. What we're hoping to do is produce a medical grade or close to medical grade chitosan, which is very expensive. And speaking of expensive, hemocyanin, keyhole limpet hemocyanin, you can purchase it commercially. It seems to me it's about $200 for 40 milligrams. It's very expensive, and we could certainly beat that by a lot. And we have data that says we have the same basic material. Collecting blood from lobsters, I can take a crate of lobsters, which contains usually 90 to 100 lobsters. In an hour's time, I can get a gallon of lobster blood, hemolymph. And then we work from there. We extract the hemocyanin And depending on whether it's going to be wet or dry, that sort of determines what the next steps will be. And just to sort of clarify this, hemocyanin, in terms of its function in lobsters and clams and crabs, shrimp, whatever, they all have it. It's like our hemoglobin. It's the oxygen-carrying material in the animal's blood, just like hemoglobin is in people. It's copper-based, whereas hemoglobin is iron-based. So it's, rather than being red, it's sort of a grayish blue color, and the color becomes more intense as the oxygen saturates. As a commercial product, we've spun this off. A couple of my former students are now selling this material as a skin cream, and it's very effective for eczema, and that's basically what they're selling it for. However, we know from our own experience, and it's sort of testing, since we don't want to get into the legalities of it, it's friends and family. And it works very well on shingles. The pain is usually gone overnight, and then it scabs over.
Some months after I interviewed Dr. Bob, when I was back in Maine shooting a different video, my dad of all people came down with shingles, and he knew who to ask for help.
For the last 2 weeks, I've been experiencing increased back pain, and so I kept getting worse. Went to the doctor, found out I have shingles. Lesions started to erupt like a rash, but really raised. And so I went to the doctor. They prescribed an antiviral medication. They also prescribed capsaicin cream, which was horrible. I put that on, and it felt like the surface of the sun. And then I got some lobster blood that Dr. Baer experiments with in a cream. And as soon as my wife applied it on the rash, it was like a cooling sensation, and then it took all that surface, that nerve pain away. I've been applying it 2 to 3 times a day. The lesions in the rash has not advanced. It looks the same as what it did. Upon application, it feels pretty good. Enough so I can work and do what I do for a living. I'm 3 days into applications. So far, so good.
By the following week, the shingles had almost completely subsided. It was Dad's first time experiencing the healing properties of this lobster hemolymph product for himself, but he relays another fisherman's experience.
An elderly fisherman here on the island, he came down with shingles, didn't really get it diagnosed until it was well along its way, and it was very painful, and he was living with it in pain for about 9 weeks. And then I found out about it, so I reached out to Bob. Bob made him some of his cream, and he sent it down. And he had tried all sorts of things for relief and could not find relief. It was in a lot of pain. And then he put that on, and he said it was a real game changer, really. It took the pain away, and it started a healing process. So it started looking better. And I think it lasted another 2 weeks, 2 to 2.5 weeks before he was better, but it really aided in his recovery and gave him much relief where he'd had no relief up until that point. So he's a big believer in it. He said it helped him a lot.
But we've tried this on any number of people, and it has yet to fail. We're trying to figure out what the proper doses are. I mean, there are a lot of questions around it, and how do we get this as an FDA-approved product with claims? And what claims? If you're saying something works, you've got to have the data that says it works. You've tested on this way on this number of people, and this is what happened. And then, of course, when we're looking at our patents are only U.S. and Canada, but I see this as clearly a worldwide application. We have direct assay that one of my students did, Matt Hodgkin. He took herpes virus, grew it up in human kidney cells. What happens is you apply your test material, in this case hemocyanin. If the cells live, that means you've killed the virus. If they don't, that means you haven't. Well, we can show that this does kill the virus using that assay. We have the most information on herpes virus and herpes viruses. I'd love to have the wherewithal to find this out, to see what other illnesses this could help with. When using parts of the lobster we hadn't before, and I kind of look at the motto for the meatpacking industry, which is, we throw away nothing. And that's the way it should be. An interesting thing that happens, if we take this lobster shell from the processing plants, instead of going to the landfill, and they're paying to have it hauled away, just by saving those disposal expenses, we're saving them tens of thousands of dollars. Some of these places are sending, they're spending $50,000 a year just to have their shell hauled away. And the blood itself, it just goes out on the floor of the processing plant, and it's actually it's quite slippery. On the cancer material, there's a lot of information on hemocyanins from other species, other than lobster, that shows that it has anti-cancer properties. We have good cell culture data that says it has anti-cancer properties. We need big pharma or big money to buy in and believe in what we have. And that's really it. It's a huge frustration to see all the evidence that we've got something that can really help people in many different ways, but we're not able to attract the backing to prove this and get it out in use for people and animals.
Wouldn't Dr. Bob's project to medically harness lobster byproducts be the perfect project for an organization like the University of Maine to back? You know, they could bring in some medical research companies in the same way they brought in wind vendors and help get this project off the ground so that it can cure many afflictions, possibly even cancer. The university's willing to go to the mat for Habib Dagger's Advanced Structures and Composites Center, pouring all of their resources into that. Wouldn't Dr. Bob's discoveries be a more helpful priority for the University of Maine than offshore wind?
This was actually sort of an annoyance. Part of my time at the University of Maine, I had a research appointment with the Maine Agriculture and Forest Experiment Station. My last couple of years, my proposed project was to develop this. And they said no.
To me, it would be a no-brainer to make an effort to cure illnesses and save people's lives, all while exponentially increasing the value of one of Maine's plentiful cash crops. Think about what the sheer amount of money being thrown at offshore wind development could yield for lobster medicinals. But apparently the university is not interested. They'd rather facilitate the filling of the oceans with turbines.
So that's why I'm doing it on my own, and that's why I made the decision to retire. I think if that hadn't happened, if I hadn't lost my experiment station project, for two reasons. One, as I see it, one is the old thing about publish or perish. Well, I was busy doing other things and I didn't publish. That was part of it, and it's also very important to bring in grant money, and I wasn't bringing enough of that. So I lost my experiment station project, and that told me it was time to retire. It's a real disappointment, but that's almost kind of the way things started. I had been at the University of Maine maybe 4 years, and I was hired to teach anatomy, physiology, some nutrition classes, and do research related to poultry. Well, I got a graduate student came along, Dale Levitt was his name, and he had this idea for a project to start looking at lobsters, and nobody at the University of Maine was doing anything with lobsters. Well, this wasn't what I was hired to do. So the administration, including the department chair and the dean, they weren't going to let me do this. So I didn't know what I was going to do about this. Well, as it happened, we just had a U.S. Department of Agriculture review, and I talked about some of the lobster stuff that I had started. And one of the reviewers was the department chairman of the Animal Science Department at UNH. And basically he says, come on down. Well, with the potential for a job offer from UNH, I could now be allowed to do my lobster research. but I really wasn't going to be well supported. I would not get any operating funds from the experiment station. Fortunately, at that time, Sea Grant was just developing, and Sea Grant and a really top-notch administrator by the name of Ron Dearborn saw the value of this, and he made sure that we got started, and because he could see the potential for impact, and because everything that I at least tried to do had impact on the lobster fishery and, again, lobster fishing families.
Maine is going to need more scientists like Dr. Bob, and his retirement from the Lobster Institute is no small loss for the University of Maine. But hopefully the right people are going to find out about his groundbreaking work and help him bring these lobster-based miracle products to market, which could actually improve the future of Maine and beyond, instead of turning it into a dystopian wasteland. If you know anyone with scientific, pharmaceutical, or angel investor connections, I'd appreciate it if you'd send them this video. The right people might help change the world for the better with the discoveries of Lobster Unlimited.